with the schedule. So it's my great pleasure to introduce Bruce Gelb, which many of you know in the field of RASopathies. He has a lot of titles. He's the Gogol Family Chair and Director of the Mintage Child Health and Development Institute, the Dean for Child Health Research at Mount Sinai, the Director of the Center for Molecular Cardiology, a Professor of Pediatrics and Genetics and Genomic Sciences. But more importantly, Bruce has done tremendous work in identifying most of the genetic causes of RASopathies, and also he's recently trying to do great work in trying to identify new therapies for some of these patients. So I'm looking forward.
Thanks so much, Pal. And it's been a wonderful meeting. I'm grateful for the opportunity to speak. First, let me get my conflicts of interest out of the way. I would say the ones that'll be relevant here are the ones with Day One and BioMarin. So although I'm giving this talk, I shouldn't be giving the talk. This person who was originally going to be giving the talk was Gregor, who's shown here on the right. This picture was taken in Dillon, Colorado after the last meeting we had 2 years ago. Katie, who's from Denver, Katie Chatfield, recommended we go there. We did. We had a great time. The smiles on our faces would be wiped away about a month later. I'll explain that in the talk, but we were happy at the moment. Unfortunately, Gregor has some family issues in Europe that have taken him there, so he wasn't able to do it, but I'll just say he has reviewed my slides and endorsed what I'm about to tell you, so that's good. Listening to Frank's talk, it occurred to me that maybe actually Gregor was a few steps ahead of me and just didn't want to talk after Frank, which is an impossible thing to do. So maybe he just stuck it to me. I don't know. Okay, so my job is to go through sort of the state of play and thinking about therapies and particularly clinical trials for the RASopathies. Obviously, we've been hearing about some of this. And so I'll try and do so. I'm going to focus mostly on the hypertrophic cardiomyopathy. I think everybody would expect that, but I'm also going to touch on some of the other things as well. And so as we start to think of where our opportunities, we've talked about these. I think some of the ones that we know are already in play are the items that I've listed here. And you can also imagine in the future that we might have opportunities for some other things, developmental delay. I particularly am interested in pulmonary valve stenosis, and even craniofacial abnormalities could be amenable to therapy over time. So the question is, how do we get there? And I think broadly there are two approaches. We're either needing to repurpose drugs or we're needing to do novel discovery of drugs. There are major advantages to repurposing, of course, although some disadvantages. I would say the key advantages are that we're taking drugs that have known safety profiles. Generally, they would already be approved by the relevant medical authorities, the FDA, EMA, for instance. They're available, and clearly, therefore, there's a faster timeline. There are some disadvantages, though, that I think are important to think about so far because they're all being developed for cancer. They are largely strong inhibitors meant to turn off the pathway. And as I'll get to in a moment, that may not be exactly our goal in the RASopathies. I think we have strong economic disincentives that certainly we've encountered along the way. And for sure, these drugs were developed for cancers and therefore are definitely not optimized for the RASopathies. On the novel discovery side, the good news is that they could be optimized for the RASopathies and therefore potentially could have fewer side effects. And one would have intellectual property rights if somebody was developing, which might help with economic factors. On the other hand, the timeline is much slower and there's enormous failure rate in discovery. And lastly, you're still going to have economic challenges in getting the backing you need to go from initial discovery to actually development all the way to the clinic. So those are our choices. And obviously, we're going to talk about repurposing because that's much further along at this point. And we've seen versions of this slide several times, I think, in this meeting, and there are many different opportunities. I'm sure there are a few things that this is now incomplete. This came from a review that I participated in. So So part of what I want to get at is how do we get to this moment? Because we've heard a fair amount about the use of MEK inhibitors and particularly trametinib, and how did we get to that moment? So here goes. Really, it was Ben Neal and Toshi Araki who developed the RAF1 knock-in model, the mouse model, and that model recapitulates nicely what we see in patients, namely hypertrophic cardiomyopathy. And they went on to then use mirdametinib and show that using that MEK inhibitor that they could prevent the mice from developing hypertrophic cardiomyopathy. I will say that that was a good experiment, but maybe not exactly anticipating what we would do clinically, since so far we have not wanted to treat patients before they develop hypertrophic cardiomyopathy. We want to make hypertrophic cardiomyopathy go away. That was not known. So at that time, and we've heard some of this as well, this is the list of the various MEK inhibitors and when they've achieved FDA approval. I'm going to tell you a story about Gregor and the first use of trametinib. Understand that at that time, the two drugs, since it was 2017 when that occurred, the two drugs that we were thinking about were trametinib and metinib, because those were the two approved. The others all came later, so they weren't really on our horizon. I should also say, and this came up with one of the questions, I believe, yesterday, the side effect profile of the MEK inhibitors is largely known. I've listed the issues here. And for the most part, as far as I'm understanding it, these are class effects. And so, pretty much we would see this from any MEK inhibitor we would potentially choose to use. So in 2017, in a 2-week period, actually Michael was first. Michael Hoffback in Germany approached me, and then Gregor approached me. They both had infants who were critically ill. One was intubated. They were both in ICU with heart failure that was unremitting, and both babies had Noonan syndrome due to RIT1 mutations. And the question was, could we try anything? And they had already failed the use of high-dose beta blockers. And so we decided as a group to go forward with trametinib, and because they have pretty wonderful pediatric pharmacology team up at Saint-Justine in Montreal, Gregor's group came up with the dosing based on what was already known. At that point, by the way, we only had the pill form. The pediatric liquid form was not yet available. And so they moved forward using the drug on a compassionate usage basis. And as people will know from the publication that came from this, that it was successful in both infants. I should mention, not only did the hypertrophic cardiomyopathy get better, but both babies had significant pulmonary valve stenosis due to dysplastic valves, and the valves got better as well. And we saw a version of this yesterday from Katie's talk. Since then, there have been a fair number of publications. I apologize if anybody has a publication that it's not included here, but this largely is the experience that's happened. And we've seen a few things. We've moved outside of Noonan to the other RASopathies. So there's now been use in both CFC and Costello. And we've clearly moved out of using it just for hypertrophic cardiomyopathy. It's been used, as we've heard, for chylus effusions. It's been used for tachycardia. And recently, it's been used for epilepsy. I think that was mentioned yesterday by Katie. And it's been used mostly in infants, but it's been used in children, and it's been used in a limited way in adults. And finally, it clearly has not worked in everybody. That there are some examples in the publications where the medicine has been used, and we have no insights into why it works for some and not for others. And as we move forward, I just want to remind people of this, because I think it's important in thinking about these therapies. And that is, the paradigm in cancer shown on the right is that we're going to use a very strong, powerful inhibitor of the RAS pathway. We're going to use it at the maximally tolerated dose. And the idea is, of course, to knock out signaling in the tumor, which is shown here in red, so a brain tumor. We're not really targeting normal signaling, shown here in gray, but we're going to hit that. And arguably, therefore, some of the side effects are going to emerge because we're turning off the pathway in tissues that are otherwise healthy. That is not what we're necessarily choosing to do in Noonan syndrome, obviously, in the other RASopathies. There we have a germline change. The gain of function is much broader. It's sort of everywhere. And therefore, going into it, arguably, one could see fewer side effects than one might in a person who didn't have a germline change. And lastly, because we're thinking we want to normalize signaling, not turn off signaling, we don't necessarily have to use the very high level of dosing. We don't have to do the maximally tolerated dose in children and adults with RASopathies. So really the paradigm is a bit different. I should also say one other thing. I think potentially for some indications, the cancer therapy, the RASopathy therapy is going to be much longer in time than the cancer therapy. So that's also a consideration. Okay. So this was the paper that Katie mentioned yesterday. This is now 30 patients who have been treated for hypertrophic cardiomyopathy. This was a review done by Gregor Cordula-Wolf in Munich, Martin Zenker, and myself. And then, as was noted yesterday, we very carefully, I think, put together controls that had similar issues but had not been treated with MEK inhibitors. There is a slight time difference because after the information about MEK inhibitors emerged, they were being used so widely. We've shown that care and survival has not improved over time up until the time that MEK inhibitors came on the scene. So I think that was fair, but it is just know that's a limitation of the study. The median treatment time was 16 months. We did have, as Katie said yesterday, 4 deaths amongst the treated. And amongst the side effect profile, we did not have any life-threatening ones, but we did have a third of the patients who had needed symptomatic treatment for skin problems, and some of them required dose reduction or stopping therapy because of the severity of the skin problems. So shown here are the outcomes, and so in red here is standard of care. The outcome measure we chose, and remember, this is retrospective, so we picked this after seeing the data, so that's another limitation, I think, of our study. But the outcome here is we're looking for freedom from death, heart transplant, or having open heart surgery to do a myectomy to remove extra heart muscle. And you can see that out of the whole group here of 31 individuals who were in the control, roughly all but a little under 20% of the patients met one of those outcome points. Whereas by comparison, in the treated group, you can see that something like only 15% of the patients hit that outcome measure, and this is highly significant. And by the way, if you want to understand the limitations of our study, read the accompanying editorial by Daphne Sue, who in an encyclopedic fashion mentions every possible limitation of a retrospective study. And so if you want to know how to interpret this, read that. Okay, she's a friend of mine. It put a little tension there. Okay. Shown here is another figure which gets to be important for thinking about the clinical trial, which is babies under 6 months of age who had the most severe form of heart failure. This is so-called RAS class 4 heart failure. And as you can see here, the numbers are smaller. We had 10 treated babies and only 6 controls. But again, we have a very significant difference in the outcome. Here, we're just looking at survival. And not shown in the paper, but relevant to the clinical trial, which is designed, which I'm going to show you in a moment, we are now including not just the babies in class 4 failure, but those in class 3 failure. So these are not as sick. And as you can see in the standard of care, shown here in blue, now you see that roughly 60% of the individuals met the outcome, whereas in comparison amongst the treated individuals, it was more like 30%. So again, slightly larger numbers than the last infant slide I just showed you, and we achieved statistical significance. So we do have a statistically different outcome using trametinib in this group of patients. So before explaining why we're waiting on NIH to fund us, let me just tell you the history of how the community has tried to pull off clinical trials till then. So starting with the 2017 experience, Gregor and I had extended discussions with Novartis and also some discussions with Roche. And in the end, neither company chose to support a clinical trial. So they were kind in offering to pay for the drug at that time. And Novartis, as people will know, had an access program that allowed us to have free drug for patients for about 5 years. That was very helpful as well. Nonetheless, we didn't have funding for a trial. And after that, and starting about 2019, Gregor, Cordula, and myself have very regularly had discussions with small companies in biotech, as well as venture capitalists who might be investing. All of these were around new MEK inhibitors that groups were developing and trying to find paths to the clinic. And in the end, we didn't get there. Along the way, and those that were at the meeting in Denver 2 years ago will remember, we did have interest from a company called Day One. They're specifically a pediatric cancer company, and they were quite interested in the RASopathies for 2 years. And 1 month after we were in Denver, they announced to us that they were pulling out of that. They had a pan-RAF inhibitor that was successful in a pivotal trial, and they decided to focus on that. Being left at the proverbial altar, we had to decide what our next step would be. And that became even more pressing because right about then, Novartis ended their access program in the US, which meant that sometimes we were no longer able to get payments, and because insurance companies weren't always approving it, we weren't able to use trametinib in all cases. And so we made the decision based on that to move forward with a clinical trial that we would fund through NIH. I should say that Cordula and Gregor have tried getting funding through Germany and Canada, where they are respectively, but that has not been successful. And so we are taking the route of trying to get funding in the US through NIH. So to do that, we are proposing to NIH a phase 3 clinical trial. We will rely on so-called real-world data, and it was mentioned yesterday in Katie's talk that Action is doing data collection. I would say that there's a parallel effort in Europe through Cordula's Center in Munich, and we're hoping that those efforts will yield at least 100 individuals who can serve as controls. So these will be infants who were not treated with trametinib, and hopefully we'll have information about their outcomes through 2 years. On the clinical side, and why are we doing it this way? I should pause and explain that. As we've spoken to pediatric cardiologists as well to advocacy groups, at this point, I think the belief that MEK inhibitors work is high enough. I personally believe that a placebo-driven trial is no longer feasible for us. And alternative designs, such as a delayed treatment trial, is also not practical here because these babies are critically ill and often don't survive that long. So we really don't have an option. So it's either, basically, we felt we had no option. So therefore, we're doing, in essence, a single-arm trial. The calculations are that we need 25 babies in order to do this. We will treat them for a year and then stop. We actually have no idea what the right length of treatment is, but from experience so far, that seems reasonable. We know some of them will relapse. The experience so far is that if you restart the MEK inhibitor, in this case trametinib, that you will get efficacy again. And so we will treat for another year if relapse occurs and study the effects of that. And then after the second year, we will stop. So that is the treatment design. We intend to include babies that have gain of function RASopathy changes with severe hypertrophic cardiomyopathy. These are babies under 6 months, and they have to be in heart failure and hospitalized. And the reason they have to be hospitalized is because that's how we're getting the data on the controls. So where we stand now are the submit. I won't take you through what happened when we applied back in November. It never got reviewed, but the new grant went in on June 11th, so about a month ago. It'll be reviewed in October and then go to council in January. And then hypothetically, it would start in April of next year. With a year of planning, we have to enroll a single patient that first year, probably towards the end of the year. I want to say the likelihood, I don't want to overpromise, the likelihood that this gets a fundable score and on a first try is kind of on the low side. This has not been an easy study section. NIH has reorganized, so it's also going to be a whole new setup. So I don't know how it's going to go at all. So TBD is the best I can say, but we're hopeful and we'll have to wait and see. I want to say what we'll accomplish with this trial. I think we'll demonstrate efficacy formally, and hopefully that will get payers on board to approve this. And we'll certainly learn more about things like adverse effects and relapse. And what we're not going to accomplish is we will not be doing dose optimization. We're taking the original dosing that we know works. I would prefer if we had endless money to do the dose optimization, but we don't. And then we will not achieve FDA approval with this rep. I want to just quickly go through other trials that are out there because I think that was my remit. And so I wanted to mention BioMarin is doing a basket trial, a phase 2 trial with vosoritide. I'll explain in a second what vosoritide is. And Noonan is one of the 3 indications. They are looking at growth, and their endpoint is change in growth at 6 months. So for those who don't know it, vosoritide is a C-type natriuretic peptide analog. And so biologically, and we heard a little bit from Frank, we know that with gain of function, you have changes in chondrocytes. This overcomes that. It's already approved for achondroplasia. And we'll see how it does in kids with Noonan. And very quickly, there's a trial listed out of Poland in Warsaw using it for HCM. We don't know much about that trial. Sanofi has another growth hormone trial in Noonan syndrome. And a colleague from Europe, Thomas Edouard, in Toulouse in France, has tried simvastatin for growth. They have finished this trial. This is a placebo-driven phase 3 trial. The results have not been announced. I got an email from him half an hour ago. He's on vacation, having a great time, but he doesn't have results to share with us at this point. So the last, do I have time for the last couple slides? I hope okay, pal. Is that I think the future is as following. In terms of MEK inhibitors, obviously we need to get funding. Hopefully we'll do that. We can continue to explore other corporate sponsorship for other MEK inhibitors. And then we can think about expanding to other indications, particularly milder ones, as we understand the side effect profile of MEK inhibitors a bit more in our patients in a formal way. There are many other inhibitors that theoretically we could use. Several speakers have spoken to that. And the question is, how are we going to pick which other ones we would try? I will give you my own thought of that on the next slide. And I think we're a ways off till we have novel RASopathy specific drugs. But I think that this is not off the question, and I at least hypothetically could lead to even better drugs for our patients. And so this is the very last slide. And so just say this is how I'm thinking about this, that on the x-axis, you have the span from the problems in the RASopathy that really are just quality of life issues. I'm not minimizing those. It's just those are different than those are life-threatening on the other end. And on the other axis, on the y-axis, is the severity of the side effects of the drugs, ranging from minimal, very reversible ones to things that are severe and irreversible. And I think we have a zone where we can feel comfortable trying drugs depending on what the indication is versus the drug and its side effect profile. But then there's clearly a zone where the risk exceeds the treatment indication. And on the other hand, we have a zone where it's unlikely that the drug would work, which is itself probably not ethical. So not there. And how exactly we figure that out, I don't have all the answers for sure. And it'll be interesting conversations as we move forward. So I'll stop there. I think I went a little long. I apologize, but here's the people I've been working with, and I'd be happy to answer any questions.
Great talk. When you're talking about dosing, do you have any indications from your animal models about whether or not we can go to lower doses clinically?
No, well, in the mice, nobody has done dose optimization. You can use dosing. There are formulas for taking dosing in mice and trying to convert that into dosing for humans. But certainly from an FDA point of view, the coin of the realm is how it does in patients. So it might be interesting to try that. I am trying to get a grant, so far very unsuccessfully, for pulmonary valve stenosis, and that would be one of the things I hope to do if we get there with trametinib. Yeah.
Hello, Keir Shields, Great Ormond Street Hospital. As somebody who is principally a clinician, but whose relationship with research could be described as peripheral but keen, how can people in my position help? How can we link in with guys like you in order to provide a patient population who can either enter your studies or be monitored or provide control data?
Yeah, so I mean, I do think that there are a couple things. One is we definitely need better natural history data for the things we're doing. The patients in the United States in particular, we are much less organized than European sites and the UK sites, and therefore it's more difficult to gather the patients to get the data. I think we all have to work together on that. And I think when we do begin to have, but we also have to be organized around that. So, for instance, we heard from Beth right before the break about, the FDA's natural history, and then we heard yesterday from Katie about natural history, or not natural history of patients being treated. And we can't burn out clinicians asking them to enter the same data 2 and 3 times. So we have to also come together as a community. And then I do think that when the times come for trials that seem relevant, that depending on your specialty, helping us get the patients to the sites that are doing the trials to make sure that we meet enrollment. We heard a lot in our keynote talk on Friday about the importance of trials being successful. I mean, having a great design but no patients enrolled accomplishes nothing, and it just increases the cost and decreases the accuracy of the trial if it extends out over 5 and 10 years for enrollment when you had actually thought it was going to happen in 1 or 2. So I think we have to work together. And that's the last thing I'll say is right now we're in an awkward position. I didn't say this part of the ugly tale, but when the trial went in the first time in November, it included London and it included 3 sites in Canada and another 3 sites, I think, on the European continent. When it went in June, it included only US sites, and that's because NIH currently is only funding things here in the US, and that is really creating problems. And I think that is not a good thing for those of us in the RASopathy space. And I hope that changes again, but that's a problem right now. Yes.
Sigh first. I mean, I thank you for the talk and thank you for the work. And this is where NF1 finds itself not uncommonly, maybe not quite as heartbreaking or exhausting. But I have a question about the science, and then I have a question about process. On the science side, in the preclinical models or the existing natural history data, do kids age out of these manifestations? So if you can get them safely to 2 years old, do they age out of the cardiomyopathy?
So the answer is depends on what you mean. So for the most severe form, which we've targeted early because it felt like we had nothing to lose, the natural history is that about 80% of them are going to die basically within 6 to 8 months. So yes, they age out because they are no more. And so our only option is to treat them early. There are definitely patients, there are a few that will not pass away. They will not get all better, but their cardiomyopathy does improve enough that they're surviving. There are other patients because the ones with the severe HCM are just a fraction of all kids with RASopathy with HCM. And it is definitely possible that we could do trials with those. The older kids that have been treated are also kids that had septomyectomies, and then the obstruction came back as teenagers, and nobody had much appetite for retreating. And the last thing I'll say is back when Novartis had the MEK inhibitor that they had prior to trametinib, they started an adult trial because they were going to work their way down, I'm sure. I have some concerns because by then there's a lot of fibrosis, and reversing the fibrosis may not be as easy or the same as reversing the cardiomyocyte hypertrophy. I hope that's what you wanted.
Yes, there's more there, but that's very interesting. The practical question, let's say everything goes fabulous at the NIH, and I'll put up all my prayers that it does, and you get funded first time. who's providing drug for that trial? And I hope you're going to say it's the drug company, because then you could expand back to your EU and UK sites through the drug company and not use federal funds for that.
Yeah. So we put in the application with funds requested for the medication. We've been in conversation. Novartis US is the only one who can fund a US only trial, and they have no development money left for trametinib, so they have declined to provide support for the trial. Sorry to disappoint you. So, and by the way, our hearts aren't broken. It's just I'm borrowing from the show Hamilton. I'm doing research like I'm running out of time. Because I feel like I am. I want to get this done. Yeah. Thanks.